All right, in this lecture, we're still going to finish up and talk about what are the 4 macromolecules, or biomolecules, or organic molecules, whatever we decide to call them, and their functions. So we've only done 2, so in this shorter little lecture, we're going to do proteins and nucleic acids. So let's start with proteins. First of all, proteins are involved in nearly every dynamic function in your body. So they are very, very involved in what we do. So involved, and they're very diverse. There's tens of thousands of different proteins, each with a specific structure and function in the human body. So proteins are composed of differing arrangements of a common set of just 20 amino acid monomers. So for these guys, for proteins, we're going to have the amino acid be the monomer. Okay. They get a little bit more complex, though. They have a few more terms, but we'll work our way through them. So probably the most important role of proteins is as enzymes. So these are proteins that serve as catalysts that speed the rate of reaction. So speed reaction rates. Okay. And regulate virtually all chemical reactions within cells. So they regulate everything. I will say all enzymes are proteins. We'll write this out. Are proteins, but not all proteins are enzymes. So we have that down. Okay, because there's all kinds of other ones. There's structural proteins, there's signaling proteins. There's lots of proteins that are not always enzymes, but all enzymes are definitely a type of protein. So other types of proteins include, other than that, transport proteins that are embedded in cell membranes, which move sugar molecules and other nutrients into our cells. We have defensive proteins. These are kind of the antibodies of the immune system. We have signal proteins, like hormones, and other chemical messengers that help coordinate body activities. So they send such signals to help coordinate. Some other examples are receptor proteins. These are built into membranes and receive and transmit some signals. So they're kind of for talking, receiving signals and sending some out. There are contractile proteins, which we find in our muscle cells. Structural proteins like collagen, which have these long, strong fibers of connective tissues. And then we have storage proteins, which serve as a source of amino acids for developing embryos and eggs or seeds. So they're going to be an energy source to get started. Okay. The function of different types of proteins depends on their individual shapes. So they have varying shapes, and this is where structure and function kind of meet. So they have some individual shapes. A polypeptide chain can contain hundreds or thousands of amino acids linked by these special bonds we call peptide bonds. This is where they get their name because this is a very special type of bond specific to proteins. And so when I have many of these bonds together, we build a polypeptide. A polypeptide, I'll refer to it kind of polypeptide. It's sort of like a pre-protein. So it's before we kind of fold the protein to have its shape. So the shape is what makes the protein. It makes that function. So the shape kind of makes that function. So if it doesn't yet have its shape, then we just call it a polypeptide, or a thing with many peptide bonds. Okay. So the amino acid sequence causes the polypeptide to assume that particular shape. The shape is what makes the protein. Okay. It's an important fact. So like this thing, if we stretched it out all the way, that would just be the polypeptide. But since it's folded and becomes this thing, this is the protein. It has a 3D shape. All right. And you can also see this. Sometimes they do the space filling model. It kind of gets a little chaotic in the space filling model. All right. Some proteins, some of those different types of proteins will be used in like spider silks to be able to create these really strong webs that can stick to things. There really is an important word that we need to know here, and I will use it quite a bit, is that if a protein shape is altered, it can no longer function. So if we mess with the shape of a protein, we also mess with its function. Okay, so the shape has to be the way it has to be for it to function properly. If we alter the shape, it's called denaturation. So when we say we denature a protein, so it's denature a protein, it just means that we're changing the shape and it no longer functions. So changing the shape might be we unravel it. It might mean that we lose its specific shape and then we'll end up losing its function. So this is actually a key part that we want to make sure that we know, is know the definition of denature. And denature really only has to do with proteins. It's the only thing. Okay, so proteins can be denatured in higher salt concentration. It'll dehydrate things if we have changes in pH and if you have a whole lot of heat. So actually cooking denatures proteins. So they no longer function, but they still have their amino acids for nutrients. But cooking it will actually denature proteins. Okay, so amino acids all have an amino group and a carboxyl group. I will tell you, when I learned these, this is how the naming schemes are a moving target. They used to be a carboxylic acid group, so it made sense that it was an amino kind of acid, but that's just the way it goes. Those are their two groups, though. They're bonded around a central carbon, so it's going to be sort of like, here's our central carbon. You'll have this amino group over here. It's got a nitrogen and some other things going on. And then we have our carboxyl group, and it's got like an oxygen, another oxygen carbon off of it. So we've got those, and these are on a central atom. There will be a hydrogen atom, which is what this is saying, and then a group which is specialized by kind of an R. So there's an R group, which is kind of like a variable. Okay, everything, all of these parts will remain the same for every amino acid, but the R group is different, and it determines the specific property of each of the 20 amino acids. It what separates each of them. It's their little identifying characteristic, but it's variable among the 20. Okay, so a little bit better picture. You can see what I'm talking about. This is the amino group. So we know it's amino because it has the nitrogen in it. I like to say the N in there helps me remember that, the nitrogen of the amino group. Then we have a carboxyl or a carboxylic acid group, if you're old like me. Okay, looks a little bit like this. We'll have our hydrogen on this end, and then there's the R group, which is variable, and will help identify which of those particular amino acids it is. Okay, so amino acids are classified as either hydrophobic or hydrophilic. So remember, hydrophobic is water-fearing, and then hydrophilic is water-loving. So either it gets along with water because it has some positives and negatives to it, or it's hydrophobic and there's just equal sharing and it's pretty kind of neutral all the way through. And so it won't get along with water, or it will. So some hydrophobic ones are like this one for leucine. You'll notice that it's just carbons and hydrogens. It's equal sharing there. That's not going to get along with water very well. Some hydrophilic ones, like the OH there, it's sort of got something going on to it. We've got aspartic acid here with some oxygens and hydrogens. So there's some little unequal sharing there, which will make those a little bit hydrophilic. Okay. Amino acid monomers are linked together in a dehydration reaction. So this is kind of a continued theme, is that a dehydration reaction is what puts these together. So we're going to add a water and, sorry, not add a water, we're going to lose the water and then put things together. So dehydration, synthesis, lose the water, add them together. So we join the carboxyl group of one amino acid to the amino group of the next one, and this creates that very special bond called a peptide bond, which is where these guys get their name. So additional amino acids added to the same process create a chain of amino acids called a polypeptide, a thing with many of these particular bonds. It's a polypeptide. Okay, so we can sort of see it here. We've got the two components of water. We're going to add those together and dehydrate it or lose the water. and then synthesize or make a peptide bond somewhere around right here. Okay. And this guy we called a dipeptide because it has two things in the peptide bond. Okay. Just two molecules involved with a peptide bond. That name gets a little confusing, not how many bonds we have. Okay, so this is a good place to take a little break if you need to. So this would be a good place to pause the video, take a look back at your notes really quickly, and then rejoin. All right, so this seems like a good time where we can do a little extra credit. So this will be your Easter egg for this particular time. Just make sure that you write this down on a separate sheet of paper and hand it to me at the beginning of the next class period, and I'll give you 5 points on your test. But I want you to give me the 4 levels of structures of proteins, and you can give me a really brief kind of description. Okay, so each of these 4, write them down, and then give me a description as we go along. Okay, so a protein can have 4 levels of structure: primary, secondary, tertiary, and quaternary. Okay, and they're going to kind of add on each other. Oops, I forgot to take that one out. But here's sort of what it looks like. So primary is really just going to mean it's the sequence. So that's all I see here is that it's the sequence. Okay, and so it's just one amino acid after another. I already forgot it. Glycine, serine, lysine, cysteine, proline, leucine, methionine, valine. That's telling us the sequence. So it's just the sequence as the primary structure. The secondary structure are one of these two things. You could just write this as that there's an alpha helix or a beta pleated sheet. That could be your description, one or the other, because that's all they are. Either they kind of spiral around like this, or they form these kind of folds, okay, which we call pleated sheets, because when you make folds, you make pleats. All right. Tertiary structure then means that we've got, in addition, it's beginning to get a 3D shape, but these 3D shapes comes from, we've got 2 plus either alpha helices or beta pleated sheets. So we've got two types of secondary structures together in here, if that makes sense. Okay, then the last one is the quaternary structure, and this is actually 2 plus polypeptides put together. I'm going to put polypep for short. But now if you can kind of see here, we had one of these for tertiary structures where it has the 3D shape with multiple alpha helices and beta pleated sheets. But for quaternary, now we have 2 or more of those polypeptides hooked together. That'll make a functioning protein. Okay, so there we go. Now let's do nucleic acids. There's not a whole lot to this one because there's really only 2. So an amino acid sequence of a polypeptide is programmed by a discrete unit of inheritance known as a gene. So we actually get this amino acid sequence of polypeptides from genes, from the DNA. That's how these two guys are related, because this is going to give the DNA, or is going to give the instructions for proteins. So if you want to, it might be helpful to know is that DNA will give instructions for proteins. All of the proteins are made because of the instructions of the DNA. Okay, so DNA is called deoxyribonucleic acid. That's where the N and the A comes from, is for nucleic acids and DNA. This is actually the type of sugar, but we'll get there a little bit later. Okay, DNA is inherited from an organism's parents. DNA provides directions for its own replication. So it can program itself to make more of itself. Isn't that crazy? DNA is a tricky little guy. It also programs the cell's activity by directing the synthesis. Okay, let's try again. DNA programs the cell's activities by directing the synthesis or the making of proteins, which is what we're getting at with this crazy thing over here saying the instructions for proteins. That's what that's trying to say. You're just getting one where I've written all over the screen this time. That's how I'm feeling today, is writing screens. All right, so DNA does not build proteins directly. It doesn't do it. It only provides the instructions. It's kind of like a recipe doesn't make the food itself. Isn't that disappointing? I feel like a recipe should make the food. Really, right? But DNA is essentially the recipe. It doesn't make the proteins directly. It doesn't make them. It's simply the recipe. It doesn't do it by itself. Okay? Disappointing, albeit, but it doesn't build it directly. So DNA works through an intermediary. It's got kind of a thing that works in between, and that's the RNA, or the ribonucleic acid. It's your little go-between. We'll talk about it in plenty more detail later, but RNA will be DNA will be transcribed into RNA in the cell's nucleus, and RNA will be translated into proteins in the cytoplasm. You can write this one down, it's a super important thing, but when we cover Unit 4 of genetics, that's when we'll really hit this stuff hard. But it's worth kind of paying attention to now while we're kind of introducing it, so it's not a surprise when it comes up later. So basically what happens is we have a gene or a section of the DNA. Okay. Then we'll do some transcription. We'll rewrite it from DNA to RNA. And both of these are nucleic acids. So they end up in kind of the same language. Then we have a translation thing happens where we actually translate it from a nucleic acid language to a protein language. And this is kind of our fundamental basis for how we talk about kind of protein synthesis and making those things. We don't have to get too hard into it now, but I'm just showing you what those are because I promise you we will get more descriptive and more detailed with those in Unit 4. So be prepared. So DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are composed of the monomers, the one-mers that we call nucleotides. And they have 3 specific parts to them. We have a 5-carbon sugar, which is ribose in RNA, and then we have deoxyribose in DNA. I'm just making a note for you. Can you see the ribose, RNA, deoxyribose, DNA? The RNA and DNA are probably the most descriptive naming of the whole thing. Okay. Let's see. There's a phosphate group and there's a nitrogenous base. Nitrogenous just is an adjective meaning it's going to have a bunch of nitrogens in it. Okay, and we'll see what that looks like. I can click to it. Okay, so a phosphate group is a kind of a weird thing. Phosphate's got a lot of stuff going on. It doesn't follow the octet rule. We gotta have a couple of weirdos out there. So we do have this phosphate group, which is attached to a sugar. And these guys will actually form kind of a backbone for it later. It's kind of the backbone for DNA. But then we'll have this kind of identifying nitrogenous base. And they sort of usually do diagrams like this. But we'll have a nitrogen base, which will be kind of our base pair. In this particular case, it's adenine. But notice we call it a nitrogenous base because look how many nitrogens that we have in it. So many nitrogens. So it just gets descriptive to say, well, it's a base with a bunch of nitrogens in it. It's a nitrogenous base. Okay? So the DNA nitrogenous bases are adenine, thymine, cytosine, and guanine, or A, T, C, and G. These will come up later, but they're just those 4. And remember, these are our nucleotides, and these are our monomers that we're talking about. Okay. RNA also has A, C, G, but instead of T this time, we're going to have U. Okay. And it's worth mentioning now, but we'll get into it in more detail later. So I will save the funny jokes that I have for that one, and there are several. I will save them until that time. Okay, so a nucleic acid polymer, or a polynucleotide, pretty descriptive, a mini nucleotide forms from the nucleotide monomers. When the phosphate of one nucleotide bonds to the sugar of the next by, oh look at that, dehydration. We lose the water and stick them together. This produces a repeating sugar phosphate backbone with a protruding nitrogen base. Let's see if I can get back here. That's what I was getting at. See, notice my little backbone here? These guys would hook on to another phosphate group and then another sugar, and so it would sort of repeat on like that. There'd be another little nitrogenous base that stuck out like that. Okay. Just we look at it. But here's sort of what it would look like. So maybe we can say this is the phosphate group, this is the sugar, phosphate, sugar, phosphate, sugar. Right? And each one protrudes out with that nitrogenous base, and that as a whole is the nucleotide. So that's how those guys kind of fit together. All right. So RNA is usually a single polynucleotide strand. It actually makes it sort of unstable comparatively because it just, it's not meant to last very long. We'll get into that later, but it's a single nucleotide strand. DNA, on the other hand, is a double helix, which has two nucleotide strands that wrap around each other. So it's got that kind of double helix structure. There's one spirally thing, let's see, and then another one with it. I have better models, or you can Google it. Either one works. Okay. The two strands are associated because their particular bases are always hydrogen bound to one another. So A always pairs with T, and C always pairs with G. Here's my trick for that one. I always say straight letters pair with straight letters, and curvy letters always with curvy letters. So that's how I remember it. Somebody else might have a pretty good one, but that's mine. Straight letters with straight letters, curvy letters with curvy letters. So descriptive. Here's a much better look at what the double helix would look like. Two strands where we've got pairing from either side. You can see the curvy letters pairing together and those straight letters pairing together. They pair. We've got our sugar phosphate backbone. We've got two strands helixed or twisted around each other. It's a beautiful thing. Okay. All right, so a quick last little bit here, the evolutionary connection. A majority of people stop producing lactase, like Mr. Royce, for example, in early childhood, and so it's not easy to digest milk sugar lactose after that. So lactose intolerant represents a relatively recent mutation in the human genome, and is a survival advantage for human cultures with milk and dairy products available year-round. So tend to find it more in, I want to say there were just like a lot of farming areas that were kind of in colder areas where this became more of a thing. But a lot, and it's mainly in white folks, just to say that is that it tends to be more in Caucasians than, or the lactose tolerance seems to be more in Caucasians, and lactose intolerance seems to be a problem for everyone else, including me. It just seems to be a higher percentage in all other, because mainly it was in these farming communities and colder areas that were inhabited by mostly Caucasians. So it's just how that kind of mutation and evolutionary thing played out. All right, so researchers have identified 3 new mutations in 43 ethnic groups in East Asia that keep the lactase gene permanently turned on. So we do have another thing where we've got a couple of ethnic groups that do have it turned on in other areas. Okay. But the mutations are all different from each other and from the European mutations. There is another mutation in the European one, which is the one I'm referring to. Okay, they appeared to have occurred about 7,000 years ago and occurred about the same time as the domestication of cattle in those regions. So interesting little kind of historical fact on that. And that's how you see in some of these areas we will see these really cool drawings that depict the domestication of cattle. All right, so with that, we are finished with this particular section. That's probably stuff that we haven't heard before. It might be new things. So if at any point in there you have any questions as you sum up and finish up chapter 3, send me an email. I'll be happy to respond and we'll get it figured out.